तक चला 10. Good morning, students. Now we'll start with another topic, that is regulation of respiration. And at the end of this topic, you will be able to get the answers and have knowledge of how the respiratory system and our higher centers, they regulate our respiration. Chemically, what is the role of HIN, what is at the end of the lecture, you will able to judge how the various respiratory centers are working, what is the role of HIN at the end of the lecture, what is the role of carbon dioxide. Then another at the end of lecture, you will be able to also have knowledge of chemoreceptors, both central and peripheral. So let's begin. Once again, very good morning and welcome you students. Okay, so we'll start with the chemical control of respiration or chemical control of breathing. So what is the role of hydrogen ion? Mainly the chemical control of respiration occurs by this. We have to mark HIN, concentration of CO2, PCO2 and PO2 and PO2. So just note chemical control of breathing, that is how the control occurs by the HIN, PCO2 and PO2. Now, all the hydrogen ion concentration and the respiratory gas composition of the arterial blood is influenced by the respiration. Breathing activities are directly related to PCO2. Okay, remember, PCO2 पे depend करता है and the HIN. So, the breathing activities are directly related to PCO2, yes, very good, and HIN, and inversely related to arterial blood PO2. Inversely related to arterial blood PO2. Okay, so what do we mean by that, Peter? Related inversely to the arterial blood PO2? Yes, that the low concentration of the O2 will trigger your respiratory center. An increase in the PCO2 will also trigger. So two sets of receptors are there. कौन से receptor play role कर रहे हैं in our chemical control of breathing? Two sets of receptors. Yes, that detect the chemical changes in the blood are peripheral chemoreceptors and central chemoreceptors. Which two? Peripheral and central. Hypoxia, hypercapnia, and acidosis stimulate respiration, and this in turn raises PO2 and lowers the PCO2 and raises the pH. So all these conditions, remember, hypoxia, hypercapnia, and acidosis stimulate the respiration and in turn raise the PO2, but lower क्या हो जाता है? PCO2 and raise the pH. Now the response to the carbon dioxide. Response to the carbon dioxide and blood pH depend mainly on central chemoreceptors. Very important, underline it. Central chemoreceptors. So सेंट्रल कीमोरिसेप्टर्स किसको इन्फ्लुएंस में है? They respond mainly to carbon dioxide and blood pH. And where they are located? They are located in brainstem. Okay. And the response to hypoxia depend mainly on peripheral chemoreceptors. So, CO2 and pH response is mainly on the central chemoreceptors, whereas for the peripheral chemoreceptors, response is to hypoxia. This is very important point. यही basic point है बेटा, यही basic concept है. Then peripheral chemoreceptors, where they are? They are located in carotid and aortic bodies. Peripheral chemoreceptors. Okay? Peripheral chemoreceptors. They are located in carotid and aortic bodies and they respond. Peripheral chemoreceptors किसको respond करेंगे बेटा? They respond to changes in PO2, PCO2 and pH in the arterial blood. But they are more sensitive to decreased arterial pH. So peripheral chemoreceptors, क्या है concept? That they are more sensitive to decreased arterial pH. That is hypoxia. So they are mainly, the peripheral chemoreceptors are mainly responding to the decreased arterial pH. Though they also sense increase in PCO2, they also sense increase in PCO2 and decreased pH. So peripheral chemoreceptors mainly they sense increase in PCO2 and decrease pH, more sensitive to decreased arterial pH. So peripheral के संग हमारा क्या mainly जुड़ेगा? Decreased arterial pH. Carotid and aortic bodies send signals to the dorsal respiratory group. Of neurons in medulla to increase ventilation. So, carotid and aortic bodies से signals कहाँ जाएंगे? Send signals to the DRG. What is DRG? DRG is dorsal respiratory group. Okay. So, peripheral chemoreceptors are mainly located in the carotid bodies and they are more sensitive to decreased arterial pH. Functions of the carotid bodies. The functions of the carotid bodies as chemoreceptors was reported by Cornel Emmons, for which he also got the Nobel Prize. These are very small. The carotid bodies, they are very small, chemosensitive organs, having width of about 2 millimeter and weight of about 2 milligram. Weight of around 2 milligram. So they are very small, very, very small. Chemosensitive organs and only having weight of 2 milligram only. Where they are present, they are located at the bifurcation of common carotid arteries. Chemoreceptors, they are located at the bifurcation of common carotid arteries near the base of the skull on the both sides. Carotid bodies, they are present at the bifurcation of the common carotid arteries. Afferent nerve. Which afferent nerve passes through the carotid bodies? Commonly asked. But that is an MCQ question also. Okay. Viva. And of course, concept to hai, to you will add this in your theory also when you are answering if you get a question on this. Then afferent nerve कौन से है? Afferent nerves from the carotid bodies travel to the brainstem is in the glossopharyngeal nerve. Glossopharyngeal nerve. This you have to remember. For carotid bodies, afferent nerve is glossopharyngeal nerve. Very important. So carotid bodies, they are small, chemosensitive organs and located at the bifurcation of the common carotid arteries. And afferent nerves is which nerve? Which nerve? Yes, glossopharyngeal nerve. Glossopharyngeal nerve. Location, here you can see. This is external carotid artery. Common carotid artery and external carotid artery. Here is the carotid sinus. And here is at the bifurcation, the carotid body is. This is the location of the carotid body. This is common carotid artery. This is external carotid artery. And this is the carotid. Here is the location of the carotid body. Draw कर लीजिए quickly. Simple diagram. Yes. Make a common carotid, then external carotid artery, then your carotid sinus, and at the bifurcation is present is the carotid body. What are the special features of the carotid? High blood flow. High blood flow. They receive unusually high blood flow, which is highest in the body per unit weight of the tissue. So highest blood flow to the carotid bodies is highest. High blood flow. It is an important feature in terms of the weight. The blood flow is about 2 liters per 100 gram of tissue per minute. Blood flow is 2 liters per 100 gram of tissue per minute, mind you. So it is having a very highest high blood flow, highest blood, which is approximately 40 times the blood flow of brain. If we compare the blood flow with the brain, so it is approximately 40 times more to the brain. They have high metabolic rate. Another important feature, they have high metabolic rate. It is about 3 times greater that of the. So 40 times more blood flow than brain, and metabolic rate is around 3 times greater than that of the 3 times greater than that of the. Due to this greater blood flow, the carotid bodies easily detect minor changes in the PO2, PCO2, and pH of the blood. So, this important characteristics enhance its own function. So, any any minor change in the values of the PO2, partial pressure of oxygen, partial pressure of carbon dioxide, and pH of blood. This will be easily detected by the carotid bodies because of the highest blood. However, in spite of greater metabolism, however, in spite of the greater metabolism, the composition of PO2, PCO2, and pH in the carotid body is virtually same as that of the arterial blood, as that of the much higher blood. So, special features of the carotid body is the highest blood flow per unit weight of the tissues, which is around 2 liters per 100 gram of tissues per minute. So just underline it. That is 40% more that of the brain and 3 times more metabolic rate than that of the brain. Hence, it can easily detect any minor changes in the PO2, PCO2, and pH. And scientists contributed, we have already seen, Connelly, J.F. Emmons, he got the Nobel Prize for it. Carotid body have 2 types of cells. 2 types of cells. Type 1 and type 2. Type 1 and type 2. These cells, they are surrounded by the fenestrated sinusoidal capillaries. They are surrounded by the fenestrated sinusoidal capillaries. Type 1 cells are chemosensitive cells. They are chemosensitive cells in the carotid. Or glomus cells. Or glomus cells. So, type 1 cells or the glomus cells. Or the glomus cells. Glomus cells are spherical in shape, having diameter around 10 micrometer. They are neuroectodermal in origin. Neuroectodermal in origin and structure resemble chromaffin cells of the adrenal medulla, and neurons of peripheral nervous system. They contain voltage-gated ion channels. They contain voltage-gated ion channels on their membrane and many granules in the cytoplasm containing catecholamines like norepinephrine, dopamine, norepinephrine and dopamine. Substance P, met-enkephalin and acetylcholine. Met-enkephalin and acetyl. These chemical substances, especially catecholamines, mainly dopamine, are released from globus cells in response to hypoxia. So voltage-gated ion channels are there and many granules in the cytoplasm are there. So type 1 cells, they are the, another name, I've told you type 1 cells, another name is, yes, students? Yes, glomus cells. And these are chemosensitive cells. These are chemosensitive cells. And they are neuroectodermal in origin, spherical in shape, having voltage-gated ion channels and many granules. So here you can see in the diagram very well. Type 1 glomus cells, type 1 or the glomus cells. Adjacent to it is the type 2, supported by the type 2 cells, and closely surrounded by the fenestrated capillaries. What are these? These are the fenestrated capillaries. Efferent axon is ninth nerve. So these are the type 1 glomus cells, type 2 cells supporting the type 1 cells, closely surrounding fenestrated capillaries, and the efferent nerve is your ninth nerve. Dopamine released by glomus cells. Dopamine released by the glomus cells acts on D2 receptors. Acts on the D2 receptors present on membrane of ninth nerve ending. So neurotransmitter कौन सा बेटा? Dopamine. And receptor कौन सा है? D2 receptors. D2 receptors is present where? Yes, on the ninth nerve ending, and triggers the action potential in the carotid sinus nerve. Now, this close contact of the fenestrated capillaries with the glomus cells helps the cells to easily monitor the changes of gases in the arterial. Easily monitor the changes of the gases in the arterial. Type 2 cells. Type 2 cells are also called sus-tentacular cells. They surround the clusters of glomus cells. They are the supporting cells. They are the supporting cells for glomus cells, as glial cells support neurons in the brain. Supports the neuron in the brain. So type 2 cells are sustentacular cells and surround the clusters of the glomus cells. So receptors हमने कौन सा पढ़ा? D2 receptors on the ninth nerve ending, and the neurotransmitter is our dopamine. Because of the presence of the fenestrated capillaries with the glomerular cells, they help the cells to easily monitor the changes of the gases. Aortic bodies. They are located along the ascending aorta and the arch of aorta. Ascending aorta and the arch of aorta. And they are innervated by the vagal afferents. Which nerve? Yes, vagus. Vagus nerve. This you have to remember, very important. So, where they are located? They are located along the ascending aorta, along the ascending aorta, and the arch of aorta, and the arch of aorta. They also contain glomus cells. They also contain glomus cells similar to that of the carotid body. Carotid body. But the response is different. Response of aortic chemoreceptors to stimuli are somewhat different from the carotid chemoreceptors. Are different from the carotid chemoreceptors. Also, aortic chemoreceptors are relatively less effective. They are less effective than carotid chemoreceptors in regulation of respiration. In regulation of respiration. So, aortic bodies are located along the ascending aorta and the arch of aorta, containing glomus cells. Aortic chemoreceptors are less effective than the carotid chemoreceptor. Than the chemical regulation carotid chemoreceptor. Chemoreceptors respond to hypoxia, hypercapnia, and acidosis. Interesting note is that the mechanism of chemoreceptor stimulation is different from the mechanism of sensory transduction as described in the sensory system in neurophysiology. So, chemoreceptors will respond to hypoxia, hypercapnia, and acidosis. The sensor for chemical stimuli is the glomus cells. Which cell? Glomus cell. So, glomus cell is the sensor for chemical stimuli which respond to all three chemical changes in the blood. It responds to all three chemical changes in the blood. Final pathway of the mechanism for three stimuli is also the same in which inhibition of the potassium channel results. So what happens is the inhibition of the potassium channel results in depolarization of induced opening of voltage-gated calcium channels. Results in depolarization induced opening of voltage-gated calcium channels that in turn releases neurotransmitters and causes stimulation of afferent fibers. Causes stimulation of afferent fibers. Okay, so there is inhibition of the potassium channels, result in the depolarization induced opening of the voltage-gated calcium channel. However, the mechanism that causes inhibition of the potassium channel by each stimulus is different. However, the mechanism that causes inhibition of potassium channel by each stimulus is different. Hypoxia. Hypoxia is the major stimulus for activation of the peripheral chemoreceptors. With normal blood pH and PCO. So major stimulus for activation is, yes, hypoxia. So with this you have to remember very important point. Very important concepts. You just mark it. Underline it. Take a star. Stimulus for activation of the peripheral chemoreceptor is hypoxia. With normal pH and PCO. Increase in PO2 above normal range does not have significant effect on breathing. It will not have much significant effect, but decrease in PaO2 less than 100 millimeter of mercury results in progressive increase in the firing rate of afferent nerves that increase the ventilation. The changes in the ventilation to alteration in the alveolar PO2 is not linear. The response becomes prominent when alveolar PO2 falls below 80 millimeter mercury. So response बेटा हमेशा linear नहीं रहेगा, है ना? Maximum when it is maximum, yes, alveolar PO2 when it falls below 80 millimeter of mercury. 80 millimeter of response is most effective. It is most effective at PO2 less than 60 millimeter mercury. Less than 60 millimeter mercury. Below this level, ventilatory responses curve is steep and almost linear. So hypoxia is the major stimulus for activation of peripheral chemoreceptors with normal blood pH and CO2. And decrease in PO2 less than 100 millimeter of mercury results in progressive increase in the firing rate of afferent. The response is most effective at PO2 less than 80 millimeter mercury. Less than 80 millimeter mercury. Below this level, ventilatory response curve is steep and almost linear. Okay? So response is most effective at PO2 less than 60 millimeter mercury, and at this level क्या होता है? Ventilatory response curve is steep. This you have to remember. Okay? So nerve traffic, impulse, arterial PO2 being 50, 60 steep, 100 and 150, we don't get a linear curve, okay? Millimeter of mercury. Nerve traffic in ninth cranial nerve. Discharge rate is maximum at lower level of arterial PO2, especially when PaO2 is less than 60 millimeter. At lower level and less than 60 millimeter of mercury. Minute ventilation, PaCO2 around 43 millimeter mercury, alveolar PO2 20, 40, 60, 80. Ventilatory response to hypoxia by hypocapnia occurs secondary to the hypoxia-induced hyperventilation. Secondary response to the hypoxia-induced hyperventilation. Mechanisms of less rise in ventilation when PO2 falls from 100 to 60 millimeter mercury. Falls from 100 to 60 millimeter of mercury. In hypoxia, hemoglobin is less saturated with oxygen. So, oxyhemoglobin is a stronger acid than hemoglobin. So, it is a stronger acid. So, with fall in the arterial PO2, the H-ion of the arterial blood falls due to less saturation of hemoglobin with blood, and the fall in H-ion inhibits the respiration. So what happens is, whenever there is fall in H-ion, inhibits the respiration. Increased ventilation due to hypoxia decreases PCO2. That in turn inhibits ventilation. That in turn, what it will do? Will inhibit the ventilation. Therefore, unless hypoxia is strong enough to overcome the inhibitory effect of decreased H-ion and PCO2 in the arterial blood on ventilation, its effect on ventilation is not formulated. So unless hypoxia is strong enough to overcome the effect, when PO2 is less than 60 millimeter of mercury, hypoxia drive becomes strong enough. Strong होती है, noted. Becomes strong enough to override these inhibitory effects. So maximum PO2 fall from 100 to 60 millimeter mercury. In hypoxia, hemoglobin is less saturated with the oxygen. Now, what is the significance? Yes. What is the significance? Response of peripheral chemoreceptors to oxygen depends on PaO2 and not on the oxygen content. Therefore, in anemia or carbon monoxide poisoning, the two conditions that exhibit reduced oxygen content but not normal PO2 have little effect on the ventilatory response. It depends on the PaO2 and not on the oxygen content. Remember, mechanism. Hypoxia inhibits potassium channels. It is inhibiting the potassium channel. The accumulation of potassium in the glomerulus cells results in depolarization that activates voltage-gated calcium channels and increase the calcium. So, क्या करता है? Increase the calcium influx. So hypoxia is inhibiting the potassium channel and there is increase in the calcium. So hypoxia increase in the cyclic AMP, inhibition of the potassium channels, decrease potassium efflux, depolarization of the glomerulus cells, depolarization induced opening of the voltage gated calcium channels, influx of calcium, calcium mediated release of neurotransmitters, increased rate of discharge of the carotid nerve, stimulation of the respiratory center occurs, and increased ventilation. So this is the mechanism of increased ventilation by the hypoxia via the loss of oxygen from the heme containing protein and decreased mitochondrial and NAD oxidation. So we'll conclude. Inhibition of the potassium channel occurs. Inhibition of the potassium channel occurs by the hypoxia. Occurs by three possible mechanisms. Yes, heme containing protein, which is closely associated with the potassium channel, and hypoxia increases cyclic AMP in the glomerulus cells. Increased intracellular cyclic AMP inhibits cyclic AMP sensitive potassium channels. Hypoxia inhibits mitochondrial NADPH oxidase. NADPH oxidase. So, to conclude, we have seen that how the chemical regulation of breathing occurs. So, at the end of the lecture, we are able to tell the role of peripheral chemoreceptors, the central chemoreceptors, where they are located, what is the mechanism, what is the mechanism of decrease CO2, hypoxia, PCO2, and the pH. How the pH is affected and the mechanism is associated, we have seen with the potassium channel inhibition. Hypoxia increases cyclic AMP. Hypoxia inhibits mitochondrial NADPH oxidase in the glomerulus cells. So I'll stop here. Okay, beta. Thank you, all of you. If you have any question, you can ask.